Have you ever wondered how a tiny seed transforms into a towering tree? The answer lies within the cell cycle. Our journey begins with the cell cycle, an essential process of cell division that's foundational to life. We have learned that the cell cycle is more than just biology, it's the blueprint for growth, repair, and reproduction of all living organisms. In our biology lab, we learn to see the cell cycle through a microscope. So first, let's just dive into and learn about the different parts of a microscope. And then we can start experimenting.
When a cell undergoes mitosis, the resulting cells wind up with the same amount of DNA as the original cell. In meiosis, on the other hand, the resulting cells wind up with half the amount of DNA as the original cell before the division. A vial of Farmer's fixative, aka 1 part glacial acetic acid, 3 parts ethanol, scissors, forceps, and scalpels, a vial of 1 normal hydrochloric acid, a dish of distilled water, a dish of 2% acetocarmine stain, microscope slides and cover slips, a light microscope, and actively growing plant roots. I'm using scallions. I've let their roots grow in water for a few days prior to the experiment. First, I'll cut off some root tips into the vial of Farmer's fixative and leave them for at least 4 hours. Overnight is great too. The chemicals will preserve the DNA in the roots. Then I'll transfer them to the vial of hydrochloric acid and let them sit for 2 minutes to soften the root tissue. Next, I'll move them to the dish of water for a quick rinse. Then another move to the dish of acetocarmine where they'll sit for 5 to 10 minutes. This will stain the cell's chromatin red and allow us to see the DNA, but you can use any DNA or chromatin-specific stain for this step. And then it's just one more move to their final resting place, a clean microscope slide with a drop of distilled water on it. I'm going to chop off most of the root and discard it, leaving behind just 1 millimeter of the very tip of the root, because this is where all of the cell division is happening and will give us our best chance of seeing mitosis. Once that's done, I'll pop on a cover slip and use the blunt end of my forceps to squash the cells. Don't be shy, they should be very squished. And finally, the moment of truth. Let's put the slide on the microscope and see what we can see. Okay, here are a bunch of squashed cells, great. Let's look around. That's definitely some mitosis going on. Oh my gosh, anaphase. As plain as day, look at those chromosomes separating. Way to go,
Now
chromosomes.
let's dive deeper into mitosis and meiosis. So what's the difference between mitosis and meiosis? Mitosis is a process of cell division where chromosomes are replicated and separated into 2 genetically identical cells. Mitosis is the process involved in growth and repair of cells. Now we divide mitosis into 5 parts in this slide. Here we start from the duplication of nuclear DNA take place and it gradually boils down how 2 new sets of identical cells are generated. Meiosis is the type of cell division that produces 4 cells with half the number of chromosomes from the parent cells. This process is responsible for producing egg cells and sperm cells. In meiosis 1, tetrads are formed, and after various steps, we get to see 2 new sets of non-identical cells that are generated. The nucleus starts to form around each new cells. In meiosis 2, spindles reform around the chromosomes of each new cell. And after 2 more steps, we can see 2 new sets of non-identical cells that are generated from each divided cell. The nucleus starts to form around each new cell again. Now, what happens if cell cycle goes awry? Cancer, this word that strikes fear into the hearts of millions, begins at a microscopic level with abnormal cell division. Understanding this process is crucial in the battle against the disease. At the heart of every cancer is the cell cycle gone awry. Cells that should stop dividing continue unchecked, forming tumors. Scientists study the cell cycle's intricacies to find weaknesses in cancerous cells that can be targeted with treatments. Breakthroughs in understanding cell divisions have led to targeted cancer therapies. These treatments, unlike traditional chemotherapy, aim precisely at the rogue elements of cell division in cancer cells, sparing healthy cells and reducing side effects. These advancements have transformed cancer treatment, offering hope and improved quality of life to many. It's a testament to the power of scientific curiosity and dedication to understanding life at a cellular level. The journey from microscopic cell division to groundbreaking cancer treatments underscores the interconnectedness of biology and healthcare. The future holds promise for even more precise interventions as our understanding deepens. Every cell tells a story, and by listening closely, scientists are rewriting the narrative of cancer. It's a vivid reminder of how fundamental biological processes like cell division are pivotal to solving some of the humanity's greatest challenges. I'm Ritik Borua, and thanks everyone for watching my Biology 110 video.